So now that we know how we're going to inhibit the heparin sulfate expression, we wanted to look at some functional responses of the endothelial cells, and we first want to look at cell alignment. So we measured cell alignment post-flow, or pre-flow and post-flow, and then we normalized the cell alignment to be from 0 to 90 degrees. So 45-degree angle would be completely random, and that's what our pre-flow static cells show. And then after flow, with our controls are shown here by the gray bars, you can see that there's significant cell alignment as expected between the non-reversing and the steady flow, because it has a mean shear stress of 15, whereas oscillatory with a mean shear stress of 0 does not show any cell alignment. So it agrees with the literature results. Now when we add the heparinase enzyme, we see that there is still significant alignment compared to the pre-flow during the steady and the non-reversing, except you can see a trend here that there is some effect of the heparinase treatment on alignment. You look at the two-way ANOVA results, heparinase treatment does cause a significant effect on the cell alignment. So our results are a little bit contradictory to those of Forbes-Dewey's group, that he showed that there was no significant difference between the pre-flow and the post-flow when you add heparinase. But we do see a significant difference, but again, we are seeing some effect of the heparinase enzyme on alignment, just not a full inhibition of alignment. Also looking at nitric oxide production, we didn't see any difference due to the heparinase treatment over an accumulation of nitrates and nitrites in the media. For any of the conditions, you can see that between steady, non-reversing, and oscillatory, you do get a decrease in overall nitric oxide, although they're not significant, the trend is there. Finally, we did a microarray where we looked at over 10,000 genes for the porcine aortic endothelial cells post-flow, and doing a two-way ANOVA with an FDR of 0.05, we found that the flow condition significantly changed over 1,800 genes, whereas the heparinase treatment had 0 genes past the FDR of 0.05. So again, it's pretty strong evidence that the heparinase treatment is not greatly affecting the gene expression of the endothelial cells. Further, if you just look at the flow conditions, non-reversing, oscillatory, when you look at these conditions, there's an abundant amount of genes that are significantly changing, and then even the change with a twofold change and having the FDR of 0.05, you have a significant number of genes that are differentially expressed. So we feel like these are comparable to the literature, and we feel like our microarray was done properly. It's just that the treatment, the heparinase treatment, is not showing any difference in gene expression. Again, this is shown by, if we look at just the treatment of control versus treatment for non-reversing, steady, and oscillatory, we can even get rid of the FDR totally and make it 1.0 and just look at how many genes actually had a twofold change out of those 10,000 genes. And you only had 15 for the non-reversing, 10 for the steady, and 4 for the oscillatory. So really an insignificant amount of genes that are being have a twofold change when you add the heparinase. We can also take a quick look at a heat map and a cluster analysis, and this is just on these 1,800 flow responsive genes. You can see that there is a very similar gene expression between the non-reversing treated and untreated. The patterns are very similar, and they cluster first, as well as the steady, control, and treated, and oscillatory, control, and treated. So all this kind of comes down to showing us that there's very little effect of the heparinase enzyme on the gene response to flow. We confirmed all the results with PCR. First, we just look at eNOS, KLF2, and E-selectin, and see that in each case, the PCR agrees with the microarray that due to the treatment, there is no effect on any of these genes. Further, we looked at the effect of flow condition on eNOS, KLF2, and E-selectin, and our results with the PCR and microarray agree with each other, and more importantly, it also agrees with what's found in the literature that shows that the non-reversing, when you compare an atheroprotective and an atheroprone waveform, non-reversing control versus oscillatory, as well as the steady versus oscillatory, that you have an upregulation of eNOS as well as KLF2. It's about a 10-fold increase, which all agrees with the literature. So we feel very good about our PCR techniques as well as our microarray results. So in conclusion, suppressing the heparin sulfate for 24 hours of flow does not substantially affect key EC responses to flow. So therefore, heparin sulfate may not be the primary mechanotransducer of these responses. Further studies identifying the species effect on the EC response to flow with continuous heparinase treatment would be valuable. Thank you.